Lesson 7E, Urine Formation. Urine, or pee as it's commonly called, is 95% water and 5% waste. The waste includes ammonia, urea, creatinine, uric acid, histamine, drugs, and trace amounts of excess salt. Ammonia and urea are toxic byproducts of the digestion of proteins in the human body. Blood arrives to the kidney from the heart to be cleaned, traveling through the abdominal aorta and entering the kidney via the renal artery. This artery will subdivide into millions of smaller blood vessels called the afferent blood arterioles, one for each nephron in the kidney. The afferent arteriole will spread out to form a ball of blood capillaries called the glomerulus, which is under high pressure due to the narrow neck. This leads to the liquid part of blood and the small dissolved material in blood plasma to pass from the blood of the glomerulus into the Bowman's capsule. Large items like protein, red blood cells, white blood cells, and platelets do not pass, but small particles like water, nitrogenous waste, salts, amino acids, and glucose do pass into the Bowman's capsule to form the filtrate. The efferent arteriole is the blood vessel leaving the glomerulus, and it branches out to make the network of capillaries called the peritubular capillaries. This vast network will reabsorb important substances from the filtrate inside the nephron back into the plasma of the blood. At the proximal convoluted tubule, sodium ions are actively pumped back into the blood using ATP. As the positively charged sodium ions return to the blood, the negatively charged chlorine ions follow. With the osmotic pressure created from the sodium chloride being pumped out of the nephron, water follows the salt and leaves the filtrate in the nephron, traveling back into the blood. Amino acids and glucose are also actively pumped back into the blood plasma from the filtrate using ATP. This active transport of particles and the osmosis of water results in nearly 100% of the amino acids and glucose, 65% of salts, and 67% of water returning back into the blood plasma from the filtrate at the proximal convoluted tubule of the nephron. The loop of Henle is the next nephron structure, and its role is water reabsorption. This is achieved through a process called the counter-current exchange. At the ascending loop of the loop of Henle, salt is actively pumped into the intercellular space around the loop of Henle called the inner medulla. As the osmotic pressure in the medulla rises, water follows at the descending limb through osmosis. The water then is absorbed by the peritubular capillaries and returned to the blood. At the loop of Henle, another 25% of salt is reabsorbed in the blood, as well as another 15% of water. So far, that's 90% of salt and 82% water returned from the filtrate back to the blood. This process of returning necessary nutrients, salts, and water to the blood is collectively called tubular reabsorption. At the distal convoluted tubule, any waste from the blood that did not pass through the Bowman's capsule due to size or other limitations will be actively transported into the nephron from the blood of the peritubular capillaries. This process of pumping hydrogen ions, potassium ions, creatinine, histamine, and drugs like penicillin is called tubular secretion. Urine will now enter the collecting ducts, where more water will be reabsorbed, before it travels down the ureter into the bladder for storage. The end result of urine formation is 99% of water, 99.5% of sodium ions, 100% of glucose, and 44% of urea, a type of nitrogenous waste, is all reabsorbed back into the blood of the peritubular capillaries. The remainder is excreted out of the body as urine, which is roughly 1.8 liters of water, 3.2 grams of salt, and 30 grams of urea per day. The blood of the peritubular capillaries will travel out of each nephron. These arterioles will leave the kidney through the renal vein to the inferior vena cava, and finally back to the heart. The blood returns to the heart as all the oxygen has been used by the kidney cells to make ATP for active transport of salts, sugars, amino acids, and other material at the nephron.